Welcome everybody to today's edition of our webinar on magnetic resonance in biological systems. Please remember that we prefer that you not record the lectures because we anyway record them, and both speakers of today have agreed to recording and will upload it to the ICMRBS YouTube channel. If you have any questions, please use the Q&A box or raise your hand and then we can unmute you. I would also like to remind you that after the talks and the Q&As, we have an unofficial part where any non-answered questions can be still answered and other things to be discussed. I also would like to point out again that there's a early career researcher magnetic resonance webinar series organized by ICMRBS. And you see the people which are organizing it, and they also welcome nominations of speakers. Without further ado, I now want to hand over to Mei Hong. Thank you.
It's my pleasure to introduce Francesca Marassi as our first speaker today. Francesca obtained her PhD from University of Toronto in 1993 in Peter MacDonald's lab. And then she went to UPenn to do a postdoc in Stanley Opela's lab. And since 2000, she has been a professor at the Sanford Burnham Institute in La Jolla. Francesca has made significant contributions to our understanding of membrane protein structure and function. She was involved in the early development of the Pysema technique, which has been really a fantastic method to get protein orientations and contributed to the analysis or the discovery of the Pisa wheel for alpha helical proteins. But I actually find that a subsequent paper I like even better because she discovered how beta sheet membrane proteins patterns should look like. I call these Pisa butterflies. It's very intriguing pattern, and she demonstrated these experimental approaches as well as how to assign these spectrum using at one point this shotgun approach. So just a lot of nice discoveries and papers, publications on membrane proteins. And today she's going to tell us something maybe just slightly different or perhaps not very different about structural insights into calcified deposits. So I'm looking forward to it.
Thank you, May. Thank you. So thank you very much. Thank you, organizers, for organizing this wonderful series and keeping us connected during this pandemic. So I'll talk to you about a topic that's pretty new in my laboratory and interesting because we came about it through our work actually on host pathogen interactions and membrane proteins. And the topic revolves around understanding how a disease called age-related macular degeneration, or AMD for short, develops. So as a way of introduction, I will just note that healthy vision depends on the functionality of the visual layers. Shown here, you see the anatomy of the eye and the structure of the various visual layers with light coming in through here from the outside, traveling through the various neurons, the photoreceptor cells, and then finally ending up at this retinal epithelial layer right here that then abuts with the blood vessels. And so the health of this structure is very important because obviously you need to see, but you need to be able to exchange nutrients. Actually, should be the other way, I'm sorry, but nutrients coming in, waste going out. So this metabolic exchange is extremely important for healthy vision. So with age, one of the things that happens is that this epithelial layer begins to atrophy. And in addition to that, there's a deposition of calcified, lipid-rich, protein-rich structures that begin to accumulate at this subretinal epithelial space. Okay, so just between the blood vessel layer and the epithelial layer that provides this immune privileged structure in the eye. And so the disease progression, the mechanism of the disease progression is not completely understood. What is known is that these deposits are a hallmark of age-related macular degeneration. And interestingly, they're also a hallmark of things like Alzheimer's disease, and they share similarities with the plaques that form in atherosclerosis. And so the resulting degeneration obviously then coincides with your vision going from perfect, shown right here, to highly blurred, to completely gone, and blindness. And it's really this major cause of blindness worldwide. So a couple of images right here showing the kinds of things that you see if you actually image the eye. So when you go to the eye doctor, sorry about that, it's a crazy dog here. So when you go to the eye doctor, you get these images of the eye that can tell whether you have AMD or not. Oh man, sorry, give me a moment to get my mouse all set up here. Okay, so you can see these images. So these yellow puncta in the eye are a hallmark of these deposits forming in the sub-RPE space. Different kinds of stainings really show these sort of spherical structures that that accumulate and end up being a hallmark of AMD. Okay, so let me just try to. Apologies for the mess here.
Then it's not mess.
Yeah, no, this is home mess. Yes, this is like a new rescue dog who's just going crazy. All right, I think I'm back. All right. So we became intrigued in this topic because our collaborator, Richard Thompson, did a very interesting experiment, which he published in 2015. So what he did was he took, with his collaborators, took basically cadaver eyes and extracted these deposits that cause blindness. And he milled them with a focused ion beam and discovered that the globs that form in the sub-RPE space are actually made of these very interesting, very homogeneous spherical structures. Okay, so these spherules have an alluring composition. They have this core stained in black here or unstained, that's primarily composed of phospholipid and cholesterol. And then around that, seen here with xylenol orange stain, is actually hydroxyapatite. So that's basically bone. It's mineralized calcium phosphate that's formed around this bonbon core of phospholipid. And then surrounding that, there's an additional layer of protein. And the protein that he identified was either amyloid beta, that's seen here with the red stain. This is now immune fluorescence staining. Vitronectin, which is the protein that I'll be talking about, stained in yellow. And also complement H. So all of these are proteins that circulate in blood. And they happen to be highly abundant in these deposits. And the discovery was really these spherical structures. And the question is, how do these form? So based on this discovery, he proposed a mechanism of formation that might begin with a little lipid droplet, shown here in black, forming as we age, because of impaired metabolism. So you can imagine that you're aging, metabolism across the layers is impaired. You start to accumulate lipids, lipid droplets, and then these become the nuclei for inducing hydroxyapatite formation, shown here in pink in stage 2. Deposition of protein on top of that. I mean, we all know that HAP, hydroxyapatite, is also a chromatography medium, so it avidly binds all sorts of proteins. And then these protospherules, let's say, or these spherules then begin to aggregate and form these deposits that accumulate and cause blindness. And going back to this, the reason we became interested in vitronectin in particular is because it's unique in forming not only the spherical structures, but also these flower-like halos that you see surrounding the particle, suggesting that it almost looks like a phase separated, a phase separation of vitronectin and hydroxyapatite, suggesting that perhaps vitronectin is actually involved or plays a role, an active role, in the formation of these spherules. So I should also say that vitronectin has also been noted in the soluble amyloid oligomers and fibrils that are associated with Alzheimer's disease. And this is a nice paper by Ralph Langen, Jeannie Chen, and colleagues, showing the overlay of vitronectin staining with this OC antibody, which is highly specific for amyloid fibril formation. These are now in Alzheimer's disease. They also did an interesting experiment in which they showed that human vitronectin and recombinant vitronectin both can aggregate and form various types of structures associated with ectopic deposit formation. So from our side, this is the only slide I'll show for a membrane protein, but actually this is the way that we came to vitronectin. So we've been interested for some time in host pathogen interactions. And we discovered that the pathogen Yersinia pestis, which produces an outer membrane protein, AIL, on its surface, actually recruits blood vitronectin as a mechanism for evading host innate immunity. Just a shout out to this recent paper that we just published, James Kent, first author, where he was able to express AIL on the surface of bacteria and then collect the cell envelopes for solid state NMR studies. So this is a CP-HSQC spectrum in black, and then expose this sample to human serum, showed that the bacteria are active with respect to being able to pulling down vitronectin, and that human serum now begins to show you changes that we're hoping to map to understand the binding interaction. So the connection here is interesting. And one of the reasons that it's interesting is because it suggests this connection between abnormal deposit formation and host pathogen interaction. And the connection is innate immunity. There's a number of host innate immune proteins that play important roles in these both these types of diseases. And it sort of connects to this idea of the immune defense hypothesis of Alzheimer's disease, okay? Thinking that there's some sort of invading nucleating agent that begins to induce ectopic aggregate formation in various immune privileged areas like the brain or like the eye. Okay, so what is vitronectin? So vitronectin was discovered back in 1967. Erkki Ruoslahti at our own institute did a lot of work showing that it contains this famous RGD motif, arginine glycine aspartate, that makes it a major integrin ligand, and indeed it's involved in, in cancer, in cell mobility, cell adhesion, but also, as I said, in immunity. It's a major component of lipoparticles in blood. So it's a multifunctional protein with all sorts of glycosylation sites, cysteine, protease sites, you name it. For a long time, the structure of this protein was unknown. Only the structure of the N-terminal domain for the residues had been determined, the somatomedin-like domain. There's a crystal structure by Zhu and Reed, and there's an NMR structure by Jane Dyson and co-workers. And this is an important part of the protein. But the remaining C-terminus was pretty much unknown and poorly understood because the sequence is complicated, it's highly repetitive. And so because of our interest in these topics, we embarked on a deep sequence analysis. We went way down in evolution and analyzed both the sequence. I compared it to the exonic structure, to the genetic structure And so what that allowed us to do was to come up with a domain organization for the protein. So it's made up of 4 repeating hemopexin-like domains, 1, 2, 3, and 4, and 2 regions that are somewhat disordered, at least predicted to be disordered, that we call variable regions. And so based on this analysis, we were able then to produce a construct that we could purify, unfold it, refold to high homogeneity, purify, and use for structural determination. So this is a crystal structure, and it's the only crystal structure I'll show you of this hemopexin-like domain. But just to show you, here are the 4 leaflets of this 4-bladed beta propeller. It very much looks like the hemopexin domain of matrix metalloproteinases that the Bertini group, Claudio Lucchina, Lucia Banci, and co-workers have been studying and have solved many structures of. And it's interesting because it contains a central channel, 2 metal ions bound by a central chloride ion in this cavity. And then the surface, like these side loops, we believe are the functional ends of the protein. And you can see that in a surface representation, there's lots of grooves and potential binding sites. And particularly, there's this acidic region right here, this red pole at the center of the protein where the metal ions come into bind. So to understand the structure, the function, of course, we turn to NMR spectroscopy. And I don't have to tell you that NMR is wonderful and it can be used to determine structure and dynamics, but it's also very useful for mapping interactions. And the ability to work with both solution and solid state NMR gives us the possibility to look at both binding to soluble metal ions, but also potentially to hydroxyapatite and lipid structures. And so one of the important things, one of the things that we noticed about the structure is this quartet of aspartates that's right at the top of the channel, that they form this chorale, highly organized chorale around the metal ion binding site. Okay, so that's shown right here. And this is really a hallmark of calcium binding protein typically. Okay, so carboxylate rich matrix proteins like osteocalcin, osteopontin, collagen, and also molecules like citrate that Klaus Schmidt-Rohr looked at in bone have these carboxylate motifs. And so in the vitronectin domain, these 4 aspartates really suggest that they might be playing a role in calcium binding. And so Kyung Shin in the lab took the protein, and he started off by doing differential scanning fluorimetry studies with various metal ions, and found that the addition of calcium in particular increase the thermal stability of the protein by a significant amount. I mean, close to 6 or 7 degrees Celsius. And so this really shows that the protein is specific for calcium binding, and calcium actually plays a role in at least stabilizing it. We turned to NMR and found that when we titrated the protein with calcium, we saw the induction of a growth of a second set of NMR signals that gain intensity with increasing calcium chloride concentration. So that's what I'm showing you right here. These are 1D slices taken from the 2D spectrum. You can see that as the calcium concentration goes up, the peak position, one peak decreases and the other peaks come up. Okay, and so based on these, on analysis of these chemical shift changes, we were able to extract an approximate KD binding affinity for calcium. And what that tells us is 27 micromolar, which is relatively low affinity, or medium, I guess. But what's interesting about it is that it is about 10 times higher than the about 3 micromolar concentration of circulating vitronectin in blood. And it's also about 30 times lower than the 1.5 millimolar concentration of ionic calcium in blood. And so what this tells us or suggests to us is that circulating vitronectin in blood is actually calcium bound. So it's a calcium binding protein in blood. And by the way, vitronectin is highly abundant in blood. Okay, so we then focused on the trying to understand where calcium binds specifically in vitronectin. And because of this conserved and repetitive structure, the signals for these 4 alanines right at the top of the rim have unusual chemical shift, have unusual proton and nitrogen chemical shift. So they're highly shielded. But interestingly, there must be some interesting electronics going on here because the 4 alanines in the spectrum completely mirror the positions of the 4 alanines in the structure, suggesting that they really do reflect the electronic structure that's right around this electronegative pole. Okay, so here they are, and interestingly, when we add calcium chloride, they shift en masse down to this position. So they maintain their register of assignment, but shift down, and the shift is highly significant. So what this really tells us is that yes, calcium really is binding to this top site and potentially replacing sodium. And most recently, we have obtained additional crystal structures that do show that calcium binds not quite at the sodium binding site, but slightly above it, and can replace it. And molecular dynamic simulation also showed this effect. The binding is very specific, so you can see that if you map various spins, various chemical shifts, amide, proton and nitrogen, C alpha, C O, C beta, they do map to this top of beta 1 right at the rim, and that pattern repeats together with the hemopexin-like repeats of the proteins. And by the way, the chemical shifts also do indicate that the crystal structure is pretty much maintained in solution. Okay, so I won't spend too much time on this, but as I said, hydroxyapatite is the mineralized form of calcium phosphate. The structure is shown right here. It's a major component of, it's bone, basically, but it's also a useful compound for the preparation of nanostructures and implantable materials. So there's a lot of interest and a lot of work done in this area, including by solid-state NMR, by many of our colleagues. And so what we did was, Kyung did, he took vitronectin and he simply incubated it with hydroxyapatite, then took down the hydroxyapatite pellet, and in this case we just bought hydroxyapatite nanoparticles, and showed that when he did that and analyzed the supernatant, all of the vitronectin was gone from it. So it's completely being pulled down by this HAP nanoparticles. And in addition to that, he also found that when he tested the thermal stability of the protein, binding to hydroxyapatite, shown in black, confers a similar extent of thermal stability as ionic calcium, shown here in pink. Whereas absorption on silica, for example, vitronectin binds to silica, does not confer such a substantial stability. And as I showed you, ionic sodium does not either. So of course, this is an ideal sample now for solid-state NMR. We turned to solid-state NMR. So in this case, we took this pellet and put it inside the rotor. And what I'm showing you here is an NCA spectrum of double-labeled vitronectin bound to HAP. A lot of overlap, a lot of work that needs to be done to improve the resolution. This is really the first spectrum that we obtained. But what's notable about it is that if we compare it to the solution NMR spectrum now of calcium-bound vitronectin, you can see that this quartet of alanines overlays with the solid-state NMR spectrum. So the shift here, the perturbations of the quartet is large enough that it can be detected even at the resolution of the solid-state NMR spectrum. So what it tells us is that it suggests that the mechanism of binding HAP is similar to that of ionic calcium. And we confirmed this by also comparing the spectra of vitronectin that was bound now to liposomes that express AIL. So this is the protein, the bacterial protein that's responsible for recruiting vitronectin to the surface. And you can see here in green that spectrum now has the alanine signal that overlays with the alanine quartet that we obtain by solution NMR in the presence of sodium only. So the combined solution and solid-state NMR data, I would say, demonstrate that calcium association is responsible for the downfield M15 chemical shifts in both the solution and the solid-state NMR spectra. And it implies that the same calcium binding site is implicated in the association of vitronectin with either soluble calcium or hydroxyapatite. And so this suggests then a mechanism, or let's say a potential role of vitronectin for hydroxyapatite mineralization and deposition. So the properties of hydroxyapatite are relevant to this question. At neutral pH, hydroxyapatite is highly stable. It's the most thermodynamically stable form of calcium phosphate. But in vivo, the surface of HAP is actually highly dynamic. And the top layer is in, the composition of the top layer is in constant remodeling, okay? So it's constantly fluctuating in response to all sorts of physical-chemical conditions. In vivo also, in vitro as well, the top surface undergoes this continuous exchange of calcium and phosphate with its environment. And as a result, it's actually less ordered than the crystalline lattice that's found in the core of the material. And so potentially vitronectin could be acting through these rim aspartates that are right here, and additional aspartates that are found on this surface, on this top, now inverted surface of the protein. And their position on the propeller surface kind of matches the interatomic spacing of the calcium ions on the hydroxyapatite 001 lattice, suggesting that they may play a role in templating the positioning of calcium ions on an HAP surface. And also, so shown here now as a cartoon of a lipid droplet, it's possible that this calcium binding activity could then coordinate the deposition of calcium ions on the phospholipids, the phospholipid head groups, the phosphates of the lipid head groups at the on the surface of lipid droplets, which are different from bilayers and liposomes. They're essentially globs of fat surrounded by a monolayer of phosphatidylcholine and cholesterol. And finally, it's possible that vitronectin could actually play a role in controlling HAP mineralization and dissolution. Just by binding to the surface, it could be expected to alter the exchange of calcium and phosphate ions. So in future experiments, I'll just give you a little bit of a flavor of what we're doing now and where we're planning to go with this. The goal is really to try and reconstitute these types of AMD spherules. And Kyung has started to prepare liposomes that he then plans to incubate with vitronectin and simulate a body fluid, basically fluid of a solution of chemically defined composition that simulates the calcium and phosphate concentrations and other ions found in blood. So what he did in preliminary experiments was to prepare liposomes, incubate it with this SBF solution, and then ask whether hydroxyapatite can deposit on their surface. And as a tool for detecting that, he has Silanol orange, which gives this beautiful pink stain in Richard Thompson's images. and can also be used to specifically image hydroxyapatite and not amorphous calcium phosphate, for example. And so what he did, what he saw when he did that was the, first of all, the formation of this bright, these are now fluorescence microscope images. So the formation of this bright corona of hydroxyapatite around the phospholipid vesicles that he prepared. And the ability of calcium apatite and calcium absorption to really form, to induce liposome aggregation and liposome fusion. So he's got really nice examples here of things like hemifusion of vesicles going on. And you can see two membranes here being flattened out and forming hydroxyapatite. So the next step is obviously to see what happens when we add in vitronectin. Does vitronectin confer HAP formation or not? And can we generate then lipoparticles that have the composition of these native deposits that form in AMD? For NMR studies and obvious potential drug discovery studies, but really the goal is try to understand the structure of these proteins. And also using NMR, both solution and solid state, we identified a relatively hydrophobic groove on the surface of the protein. And so that's highly suggestive of potential lipid binding. And so, of course, these are experiments that can be done, in this case with solution NMR, but also with solid state NMR once we get the preparation of lipid droplets and other lipid structures done. Okay, so with that, I'm going to close and acknowledge the people who really pretty much did this work. In addition to that, multiple collaborators work with us on various aspects of the project, and particularly Greg on host pathogen interactions, one pill, and desimulation, Bernard on the crystal structure. He's now at WEHI. And Richard Thompson at University of Maryland for the fluorescence. And thank you for listening.
Thank you, Francesca, for this very interesting talk. So we're open for questions. I guess while people think about the questions and type it in, I'll start. On your slide showing the model, the three different mechanistic possibilities for the vitronectin, I'm curious, you mentioned the the middle one, the lipid droplets aspect, that these lipid droplets are rich in phosphatidylcholine and cholesterol. I'm wondering if phosphatidylserine plays any role in this, because that lipid is known to, like to interact with calcium.
Right. So that's one of the things that we're going to have to explore now, for sure. So lipid droplets are actually rich in neutral lipids. So they are rich in phosphatidylcholine, cholesterol. There are some instances that induce the expression of phosphatidic acid on the surface of lipid droplets. And that's that has a high affinity for calcium as well. So that's one of the things that we're going to be exploring. But yes, that's definitely something that we'll have to do, explore the lipid composition of these structures as we try to reconstitute the protospherules and go from there. Yeah. Absolutely. Thank you.
Yeah, maybe a follow-up question is it known what's the orientation of the protein relative to the surface of the lipid droplets and relative to the hydroxyapatite surface? Is this a pretty sure model, or is there already data out there?
Well, okay, so relative to the hydroxyapatite surface, we're pretty sure that this is the orientation, just because we identified the calcium binding site right here. And the solid state NMR data showed that the same sites are involved in binding hydroxyapatite bound calcium. So I think that this is the orientation of HAP binding. With respect to the lipid, that's an open question. That's something that we have to look at. And as I said, vitronectin is a major component of, I think it's HDL. And so there's a lot of interest in trying to understand how it associates with lipoparticles and, in this case, lipid droplets. Yeah, so stay tuned, hopefully.
Okay, so the questions are coming in. So Grace Royapa asks, how many calcium can bind the cavity?
So it looks like it's one calcium. We now have a crystal structure. We had done some MD simulations and calculations in the original paper showing that this top site is accessible to calcium, but this one is not. And we now have crystal structures that confirm that, that proposition. So probably one right here. Not right here, actually, slightly higher than that, and an upper position from that.
Yeah. Marcus Cardoso asks, I would like to know if the authors considered the change in chemical shift in analyzing the NMR binding calcium. You already
Yes, we did. We did. So we use solution NMR spectroscopy, and the binding is not in fast exchange. So we cannot do a perfect tracking of chemical shift changes, but we had to resort to looking at the intensity, measuring the peak intensity as a function of the addition of calcium, and that allowed us to then determine a KD of around 25, 27 micromolar.
John Lan asks, do we know if the concentration of the spherules in the drusen is changing, normal versus diseased drusen?
Yeah, I mean, that's a big question in the field. So these spherules, I'm not sure what you mean by concentration, but so the drusen or the deposits are actually made up of spherules. So they do appear to be the major components of drusen, but it is true that, so people have noted, depending on the stages of the disease, that there are other kinds of structures that can form as well. So that's really a question for the eye doctors, but I think it is an important question in the field.
So what Masevsky asks, will lanthanides bind at the calcium site? They're often used as calcium mimetics, and they have interesting magnetorescence properties.
Yeah, thank you. That's one experiment that we would like to do, actually. We haven't tested it yet, but it's on our list of things to do, and that would certainly be a useful probe.
Okay. And Qiang Lu Fan asks, what is the biological importance or application of knowing this mechanism in the future?
So I can tell you one of the things that we're trying to do, okay, why we're trying to reconstitute this protospheral artificially, is that if we can understand what role vitronectin plays in the spheral formation, we could then potentially have a handle for drug development. Can we then identify compounds that interfere with this orchestration of HAP formation? That's the obvious translational role for this. Potentially also you could think of developing probes based on vitronectin for diagnostics, for diagnosing the disease. And then obviously just from a purely scientific standpoint, you want to fundamentally know what the mechanism of formation is. Once you know it, you could then ask things like, well, what is it that makes vitronectin go to this site? What about A beta, by the way? What about factor H, all of these other immune proteins? I mean, that's obviously very interesting, right? We're just looking at one aspect of it.
Okay, so I'll put a couple more questions into the later informal session. In the interest of time, I think we should move on to the next speaker.
Thank you.
A pleasure for me to introduce Katherine Henzler-Wildman as our second speaker. So Katie was a postdoc in my lab from 2003 to 2008 and started her scientific journey at Cornell University. As an undergrad, moved on to University of Michigan for a PhD, and then right out of the postdoc at Brandeis University, became an assistant professor at Washington University School of Medicine, where from 2008 to 2014, and then got promoted to associate professor. In 2015, she moved to the University of Wisconsin in Madison, where she's now associate professor and the co-director of the National Magnetic Resonance Facility. So I'm sure quite a few of you have interacted with her. So I must say that Katie was an outstanding postdoc in my lab, and that I was very, very fortunate to have the privilege to work with such a brilliant and passionate scientist. I remember my colleague Chris Miller telling me that in your lifetime as a PI, you only have a few of those jewels in your lab, and if you have it, you have to embrace and enjoy every moment. And I'm glad that I did that with you, Katie. So Katie really worked on very fundamental question about the hierarchy of the time scale and length scale in protein dynamics, and linking that to the importance in enzyme catalysis. And what I really want to emphasize that she was never afraid of doing the next thing I asked her to do, crazy things she'd never done, including single molecule Fred experiments, right? She came in my lab. I had done only solid state NMR, did a lot of solution NMR in my lab, but also, you know, as I said, was not afraid to add new biophysical methods. And then in the last couple of years, we sort of brainstormed to use the power of solution NMR to actually go back to work on membrane proteins. And I said, because in solution NMR, you can do anything, you can look at enzymes doing catalysis. Would you be interested to look at a membrane transporter, a multi-drug transporter, EMRE, in action when it's actually transporting, right? And again, it needed somebody ambitious like Katie to say, sure, let's do it. And so she not only did that, but also developed a method for the first time to use bicels and not what everybody else used back then for solution and then more detergents, to actually look at the drug transport in action. It was essential because in detergent, we found out it's actually not functional, right? So not surprising, she got job offers and took that project and really just developed over the years just a beautiful, beautiful you know, story, which she will tell you, not just to use NMR for structures, but actually use NMR where it's really powerful, mainly to look at mechanism. And now lately, going that extra mile to ask, by understanding the mechanism, can we actually use it for something really, really translational, which is avoiding development of drug resistance. So I'm very excited because I don't know all the data, what you have, but I think that next step to really understanding mechanism to develop that, of course, one of those stories we have heard already was the new structural model between the fluorinated substrate and you know, between the two ladies you see on the screen here between May and Katie, but she has more in the tank. So thanks again for giving us a talk here.
Thanks, Doro, for that lovely introduction. And I would like to also thank the organizers for the opportunity to present the work today. So let me just give this a section. Okay. I'm going to start, obviously I'm talking about EMRE today, and this is a little cartoon model of the transporter. I'm going to start by reviewing some of our older data because this protein, while it was originally proposed to be the simplest model system for understanding secondary active transport, has instead become one of the most complicated and broken most of our ideas about how proton-coupled transporters work. So this, let me just get this out of the way. This is a plate-based assay for looking at drug resistance in bacteria. So these are E. coli that are expressing either the wild type EMRE or E14A, which is a point mutant that's fully inactive. And these plates have a relatively high concentration of ethidium bromide. And normally E. coli without a functional drug transporter cannot grow on this concentration of ethidium bromide. It's a pretty toxic compound. But when you have functional EMRE, then it confers resistance and your bacteria can grow. And so this is one of the standard assays that's been used to characterize the function of these small multi-drug resistance transporters. So we know that EMRE can confer resistance to toxic polyaromatic cations. That's well established, and there's no dispute about that. EMRE is found in the inner membrane of E. coli. It is an E. coli protein, and its activity is dependent on the proton motive force. So it imports protons in order to drive efflux of the toxic small molecules. And what's this? There we go. So here's the structure again. This is an older crystal structure. The resolution of this structure leaves a little bit to be desired. If you've ever looked at this PDB, they only deposited C alpha. It's one of Jeff Chang's crystal structures from 2008. But it can give you an idea of the model. And if you overlay the location of the helices with the older 3D cryo-EM structure that's very low resolution in lipid bilayers, they basically align. So sort of the fundamental organization of the protein is relatively accurate. What you see here is that there is tetraphenylphosphonium here bound in the core of the protein, and it does function as a dimer. And the important thing for understanding EMRE is that it is an anti-parallel asymmetric homodimer. Now, when you have a transporter, it always has to be closed on one side of the membrane, and this is so you don't have diffusion undoing all the work you've done in uphill transport. And so as soon as you have the two halves, the two subunits of your protein inserted in the membrane in opposite directions, and then you close on one side, now you can see the asymmetry that must be inherent in the structure. These loops are exposed to water. Here, these loops are packed against the other half of the protein, and they block water access into the transport pore. And I see my glutamate has gotten moved here. There should be a glutamate here on each subunit. So there's a glutamate 14 in transmembrane helix 1, and this is the key residue for binding both protons and the drug substrate. And we know EMRE combined +1 and +2 cationic substrates, and it combined 2 protons. So based on that, a mechanistic model was proposed where you can either bind 2 protons or you can bind the drug, but not both at the same time. And this competition is important for antiport. So if you could bind drug and proton simultaneously, and the proton could then interconvert from open in to open out, that would move both the drug and proton across the membrane in the same direction, and that would be symport. In this model, you can either bind proton or drug, and then when substrate's bound, it can alternate access, switch from open to one side to open to the other, and that's what moves the substrate effectively across the membrane from one aqueous compartment to the other aqueous compartment. Here, the idea is that the protein cannot interconvert empty. It can only interconvert when bound to proton or drug, and this gives you stoichiometric coupled antiport. And the data supporting this back when it was proposed was relatively simple. For a lot of these proton-coupled transporters, you see this bell-shaped curve if you look at the pH dependence of uptake into liposomes. And the idea is that at low pH, proton wins this competition, so you oscillate here, but there's no net flux. And at high pH, drug wins this competition, and you oscillate here, but there's no net flux. And only when you're close to the PK of the active site glutamate will you have net transport. And if you have a pH gradient providing a proton motive force, then that will drive flux of the drug substrate against its concentration gradient. So that's great, but if you think about this, this is a relatively crude readout. And so we set out to use NMR to directly look at some of these steps. The dynamics here for alternating access, the competition between drug and proton, because NMR actually gives you one of the more direct ways to read out proton binding. And ideally, we wanted to sort of prove this model to get estimates of rate constants for these different steps, be able to do kinetic simulations, and provide a more quantitative comparison with liposomal flux. Instead, what we've done, and so here's just the cartoon model that you'll see versions of later in the talk. Instead, what we've done is sort of shown that there are major flaws in this model, and that's caused us to sort of rethink what this transporter is really doing. So this is just to orient you, this is the Tirozzi HSQC of EMRE bound to tetraphenylphosphonium in an isotropic bicell. And because it has this asymmetric dimer structure, we have a set of peaks from subunit A and a set of peaks from subunit B, because each of the two monomers in the homodimer have a different conformation. And then we can do TROZ selected ZZ exchange experiments. And what you'll see, so in black is the spectrum I showed you before, and in red behind it is the ZZ exchange experiment. You get these cross peaks because it turns out what's happening is that this AB dimer is going to a BA dimer. So basically the two halves of the dimer are swapping conformations. And so when you integrate your peaks to get the relative populations, you get 50-50 populations because these are just representing the two halves of the dimer. And then when it interconverts, now AB goes to BA, and now you're open to the opposite side of the membrane. This mechanism is fairly elegant. It was actually proposed when the crystal structure was published. No experimental data, just wouldn't this be a really elegant mechanism? And then this data really showed that that's actually what's happening. Now, when you analyze your ZZ exchange data, because this is a TROZ-ZZ exchange experiment, and we have, again, those loops that are flexible and exposed to water in one subunit, and then more rigid and packed against the other subunit and protected from water in the other subunit, the typical assumption that your intrinsic relaxation rates in state A and state B for exchange of a protein between two folded conformations is not true. And so you actually have to take account of this differential intrinsic relaxation. And we did that in the original publication, but for the rest of this talk, I'm going to show the data using this peak ratio method that was published by Art Palmer's group, which cancels out that intrinsic relaxation rate to first order, and so really simplifies the analysis. This is nice because you can immediately see, so each color is a different amino acid in the protein. This allows you to immediately see that all the residues are sensing exchange on the same time scale. This is also nice when I talk to non-NMR audiences because it's really easy for them to follow this simple of a plot and understand what our data is really showing. Okay, so EMRE is interesting to us because it is a promiscuous transporter. So it's a multi-drug transporter, and it recognizes very diverse substrates. And one of our early discoveries was that even when you just switch what's in this fourth position here to different substituents, we get differences in the alternating access rate of 3 orders of magnitude. We actually used a different experimental technique to get that rate because it's too fast for ZZ exchange. But I want you to remember that the alternating access rate can vary over 3 orders of magnitude just with relatively small changes in the bound substrate. And what correlates best actually with these different rates, if you think about what are the molecular properties of the small molecule that might cause these different alternating access rates, log P is really what seems to be most important, the best correlation. And so hydrophobicity of the substrate seems to play a role. Okay, so that is looking at the drug side of the transport cycle, and then we turn to the proton side of the transport cycle. And this is when we started getting into trouble. And I'm not going to go through all the data here, but basically we found that EMRE could have unprotonated, singly protonated, or doubly protonated states, and all of them could interconvert. And this is not just based on our work. Nate Trasleth has also done pH titrations and dynamics and also sees that even the empty transporter can interconvert. So now we have problems with our mechanism, right? Because the model was that you cannot interconvert because that could cause proton leak, that could cause all sorts of alternative pathways. So then we started looking at this drug-bound state again and asking, well, can you bind drug and proton simultaneously? This is sort of hearsay for the transporter community because that could potentially lead to symport by an antiporter. And so it raises all sorts of questions. Here is the pH titration. I just love to show this data because it's such a beautiful set of titration spectra. And if we focus just on alanine 10, because it's nicely resolved and close to that critical glutamate 14, what you can see is that alanine 10 in subunit A no longer titrates when we have drug bound. But alanine 10 in subunit B does titrate, and we have the expected pKa shift when we mutate that glutamate to an aspartate, indicating that we really are watching protonation of that glutamate and not another residue. And this pKa is quite close to neutral pH, so it's potentially biologically relevant. So now we have this interesting question. And if we look at where we see the largest chemical shift differences with pH, we see that they're all localized to subunit B. And then if we do a drug titration, what you can see the time scale for drug on-off is in the slow exchange regime rather than proton on-off, which is in the fast exchange regime. So we have very different spectral signatures. Then you see that it's primarily on monomer A, but also some residues on monomer B. And this is TM3, so the TRP63 is in TM3 and is one of the key residues for recognizing substrate. Okay, so now we've shown that you can bind drug and proton, and probably to a reasonable population under physiological conditions, but this is only a problem mechanistically if the transporter can interconvert and alternate access from one side of the membrane to the other with drug and proton bound. And so we went back and did the ZZ exchange experiments again at different pHs, and what you can see is that at high pH, where we're predominantly in the drug only state, or at low pH when we're in the drug and proton bound state, we have rates of alternating access that are experimentally indistinguishable. So now we have this much more complex mechanistic model, and this raises all sorts of questions. How do you get coupled transport? Why don't you have proton leak through some of these other pathways? Why can't you do symport, which would be a pathway like this? And so in the transporter community, they said, oh, well, this is all well and good that you can see these states by NMR, and kinetic simulations show that they, in theory, should be possible, but we never said they're excluded, right? You can have leak pathways, you can have slippage, and what I wanted you to think about is, what is leak and slippage really a good description of these alternative transport pathways if there are conditions under which those pathways become the dominant pathway and cause a completely different phenotype in vivo? Okay, now, since this is the most controversial state of the protein, we're going to take a little bit of a detour and talk about this structural model. So despite the fact that EMRE was one of the first crystal structures of a transporter published, it's not a great crystal structure. It's 3.8 angstrom resolution. Only C alpha was deposited because it's not a great, the electron density is not ideal. And we really want a better structure, but it's been over 15 years and there hasn't been progress with crystallography. The reason is that this is such a small protein, so nothing really sticks out of the membrane to give you good crystal contacts and give you a good, good packing. The other problem is that it's really dynamic, right? We have these fast alternating access rates. In the absence of substrate, there's huge internal dynamics on a variety of timescales. It's just a very flexible and mobile protein. So we've got some challenges. First of all, it's too dynamic, but we really want a better structure so that we can see how it interacts with different substrates and what's the difference between a drug-bound or a drug and proton-bound state. So we set out to find a mutant of EMRE with reduced dynamics that still bind substrate, because that would, in theory, prevent, right? If you have a mutant that cannot alternate access, then you could still bind substrate, but not transport. And so this gives us an easy way to screen. The other thing we wanted to do is take advantage of EMRE's substrate promiscuity. We can switch which substrate we're using. So if we use a fluorinated substrate, then we can use the fluorine proton redoor experiments that Mei Hong talked about in the seminar series recently to get distance restraints directly between the substrate and the transporter. Okay. So the first thing we did is screen for mutants of EMRE that were functionally defective. And so here you see a residue that's functionally unimportant. It can confer resistance to methyl veologen, acroflavine, and ethidium. And we mutated all of this position to valine, alanine, and glycine. And we did that throughout the protein. If you mutate the critical E14 residue, then it cannot confer resistance to any of the substrates. And then there are interesting residues like serine 64, which I'll talk about, where depending on what you mutate the residue to, sometimes it can confer resistance and sometimes it cannot. Okay, this is a collaboration with Chris Tate at the LMB. And so 7 mutants were interesting, where we had this ability to bind substrate, but defective transport. And so the hypothesis was that these would be defective in alternating access. However, when we actually started to take NMR spectra of these 7 mutants, we discovered that only one actually had reduced dynamics. And it turns out that the others are most likely altered proton drug couplings, shifts in the pKa, which would make them non-functional in vivo, or increased leak or altered drug specificity profiles. So these are the spectra of S64V EMRE. And purple is bound to tetraphenylphosphonium, and the teal spectrum is drug-free. And the first thing I think you'll notice is that the quality of these spectra is stunning for an 8-transmembrane helix membrane protein in an isotropic bicell. That these are really beautiful symmetric peaks, that there's really nice intensity across all of the peaks. And what we see is that this mutation seems to really suppress the internal dynamics of the protein on a broad range of timescales. Okay, so this is great from a structural perspective, but you need to make sure that it can still bind substrate with comparable affinity. So here's the binding affinity from ITC experiments for wild type versus S64V for 3 of those tetraphenylphosphonium derivatives, and you can see that the affinity is always within a factor of 2. This is basically identical. Remember that different substrates, we have orders of magnitude difference in affinity, so a factor of 2 is nothing. So now we have a mutant that has suppressed dynamics, but preserved substrate binding capabilities. And here's the data showing the suppression in dynamics is roughly an order of magnitude for the drug-bound state and 2 orders of magnitude in the absence of a drug substrate. The other thing we started to wonder about and to look at is what's going on at position 64. Right, so we mutate this amino acid from the native serine to valine, and we have almost no difference in binding affinity, but this 10-fold suppression in alternating access. So we did a series of substitutions at that position, and we can see again, there's much less variation in affinity, but significant variations in the rate of alternating access. And the interesting thing is that the size and the hydrophobicity of the residue at position 64 seem to correlate with the rate of alternating access. And this is reminiscent of what we found when we were looking at the different tetraphenylphosphonium derivatives, so the different small molecules. Remember there, log P, or hydrophobicity, was what was really important in determining both the affinity for the transporter and the rate of alternating access. So we're starting to see some themes here of the sort of physicochemical parameters that might affect how alternating access rates are regulated in this transporter. Okay, so now we have our dynamic suppressed mutant. Now we need to switch substrates to a fluorinated substrate so that we can use fluorine redoor experiments to get precise distance restraints. So first, we need to verify that this fluorinated substrate is actually a substrate of EMRE. And this was synthesized for us by our collaborator, Jim Davis, at the University of South Alabama. So these are a new assay in our lab. This is solid supported membrane electrophysiology. And we like this assay for liposomal flux because it doesn't require a radioactive or fluorescent substrate. So you can use any substrate, which for a promiscuous transporter, this is really useful that we can switch substrates at will. And it's much more high throughput than some of the more traditional assays that we've used, and uses a lot less material than, say, a direct pH readout, which we've also used in the lab. So in this assay, we reconstitute EMRE into a proteoliposome, and then you support, those are on a supported bilayer in the instrument. And what you do is you flush different solutions on the outside of the liposome to create gradients of proton and your drug substrate. So here in each case, we're creating a 2-fold inwardly directed proton gradient. And then through capacitative coupling, we can monitor the charge flux across this membrane and read it out through the SSME instrument. So as we start out, if you have a proton gradient and no drug gradient, then the proton gradient should drive F4-TPP out of the transporter. Traditionally, EMRE has been thought to have a 2-to-1 stoichiometry, so 2 protons for each drug substrate. And this is based primarily on the binding stoichiometry, assuming that the fact that we know you can only bind 1 drug substrate, but you can bind 2 protons, the assumption is that the binding stoichiometry is also the transport stoichiometry. There's some flaws in that reasoning, but if you have coupled 2-to-1 antiport, then 2 protons in and 1 + 1 substrate driven out would result in a net movement of +1 charge into the liposome. And so if we compare functional and non-functional EMRE protoliposomes, you can see that we do have charge transport with the functional transporter driven by this proton gradient, and there's almost no charge transport when you have a non-functional EMRE reconstituted into the liposome. If we create an outwardly directed tetraphenylphosphonium gradient, then this is consistent with antiport, right? This would drive F4-TPP out and proton in. This would also drive transport the same way. So this should just enhance coupled antiport. And indeed, we see increased flux when we have an outward-facing gradient. If you have coupled transport of F4-TPP, then if we reverse this gradient, and this gradient is larger than the proton gradient, now we should reverse the direction. This should actually drive protons uphill if we have a sufficiently large tetraphenylphosphonium gradient. And indeed, that's what we see. We see reversal of transport under these conditions. So this tells us that F4-TPP is actually being transported in a proton-coupled manner by EMRE. Okay, so we verified that this is a substrate transported by EMRE. Then we can look at the difference, chemical shift difference. So these are from solution NMR, and this is the amide chemical shift difference between drug-free and F4-TPP bound EMRE. And the subunit A chemical shifts are shown in the red color and subunit B in blue. And what you can see is that we still see larger chemical shift differences on subunit A. So remember, with tetraphenylphosphonium, we said the drug preferentially interacts with A and the proton with B. And so that's exactly consistent with what we see here. So then we use this combination of the F4-TPP and our S64V EMRE mutant and reconstituted into lipid bilayers, into liposomes, and sent these samples off to Mei Hong's group, and they did these proton fluorine reador experiments, reading it out through the HNH spectrum. So we have a proton-nitrogen correlation so that we can resolve many of the residues. And what you'll see is that now we have much more quantitative data showing this asymmetric interaction of the fluorotetraphenylphosphonium substrate with the 2 subunits of the EMRE homodimer. And when we look into this active site, so here you can see E14A, this is the glutamate that is protected from protonation and deprotonation when substrate is bound, or E14B, this is the residue that can still bind a proton, interact differently in this cluster. The other thing that's interesting is that we already knew from mutagenesis that a lot of these aromatic residues were important for substrate binding. If you mutate these, you change the substrate specificity profile of EMRE. And in particular, we know TRP63 is important. And here you can see that TRP63 from both subunits is actually relatively close to, here's another perspective, TRP63 is relatively close to the tetraphenylphosphonium, where some of the other residues that have less strong phenotypes are not as close into the substrate. So this actually makes a lot of sense with what we had sort of inferred from mutagenesis. But given the ability of this protein to defy all our expectations, it's really important for us to have these quantitative distances. And this now forms the basis for us to move forward and start to look at what's the difference between drug-bound or drug and proton-bound. So these experiments were actually all conducted at low pH. So these distance restraints are actually for the drug and proton-bound state of the transporter. Okay. So now I want to go back to the mechanism, because this is where we ran into a lot more surprises. So I said in this mechanistic model, we have issues because the NMR data suggests that our transporter should be capable not just of 2 proton to 1 drug antiport, but also of symport of drug and proton uniport, or drug uniport. And so these sort of sub-pathways are illustrated here with the different colors. This is really important to understand because antiport leads to resistance, right? The proton motive force is inwardly directed in bacteria. And so the protons will come in down the PMF, and that will drive efflux of drug, reduce the intracellular concentration, and confer resistance. However, if you have SymPort, now proton and drug are moving the same direction. That's going to actively pump drug into E. coli. So that should confer susceptibility. If you have drug uniport, well, all known substrates of EMRE are cationic, and so the negative inside membrane potential, it's about minus 190 millivolts in E. coli, that will drive the cationic substrates into bacteria and also cause concentrative uptake. So that should also cause susceptibility. And even if you just do proton uniport, that will allow protons to run down the PMF in an uncontrolled manner, and PMF rundown should also confer susceptibility. So any of these alternative pathways should result in a very different biological outcome rather than resistance. So this is really a big question, right? Could you actually see this phenotype in vivo? And I will preface this by saying that only a couple dozen substrates had ever been tested for transport by EMRE. And this is this promiscuous transporter, which we know can transport a huge variety of compounds, has not actually ever been subjected to a high throughput substrate screen. So this is our question. And what we discovered is that we can indeed see both phenotypes in vivo. So let me first show this, and then I'll talk about a larger screen. So this is a microplate growth assay. So here we have E. coli that's expressing either functional transporter, a point mutant that is non-functional, or empty plasmid. And this is because with membrane proteins that are being expressed off a plasmid, you always worry that you're making so much membrane protein that it's causing stress on the cell, and that could cause a growth defect. So here, if we only have media, you can see that all of them grow equally well. And so there's no negative impact simply from expressing the EMRE transporter. And we've previously shown that EMRE is expressed into the membrane and properly folded. So then if we have ethidium in the media, and these are liquid media assays done in microplates, then if you have functional transporter, the cells can grow because EMRE confers resistance to ethidium. But if you don't have a functional transporter, either because you have empty plasmid or non-functional transporter, then the cells cannot grow. However, we have now discovered substrates where if you have functional transporter, there's a growth defect. So expression of functional EMRE is detrimental to growth of the cell. If you do not have functional EMRE, the cells grow just fine in the presence of this substrate. But if you have an active transporter, the cells do not grow nearly as well. So this is susceptibility. So we did a larger screen. This is actually a biolog assay. So here we're not actually reading out growth. We're reading out NADH production as an indicator of cellular respiration. And this assay is, we did this one first because it's about 200 compounds. It's easy to set up. They have a sort of a chemical data set designed for bacteria. And the assay is set up with this particular 200 compound chemical screen in order to avoid effects of intrinsic fluorescence. You have to remember that this is nominally a polyaromatic cation transporter, and a lot of those compounds have intrinsic fluorescence. And so some of those assay readouts can get a little complicated. What you see here is the NADH production by wild type transporter expressing cells versus the non-functional E14Q expressing cells. So this is intrinsically an assay that's run as a difference between functional and non-functional transporter. And if there's equal NADH production by both, then you get this yellow overlay. But if the wild type has much higher NADH production, that indicates higher cellular respiration. And so this would be resistance, that the functional transporter allows the cells to grow and respire, and non-functional transporter does not. Whereas in this case, you can see that now the non-functional transporter, the cells are growing and respiring just fine, but when you have functional transporter, they're dead. So we did the screen for EMRE, which is an E. coli homolog, for the Staph aureus, one of the Staph aureus SMR homologs, and also for an SMR homolog from Francisella tularensis. And what you see here is that these in black is the number of resistance substrates that we identified from the screen, and in red is the number of susceptibility substrates. So there's a couple points I want to make here. First of all,
our organizer said you're running late, so if you want some questions.
Okay, this is the last slide, I think. Sorry. So the number of resistance and susceptibility substrates is essentially comparable, and that there are some substrates that are shared between different transporters and others that are unique to each transporter. So this is the sort of end of the talk where we have some interesting phenotypes to follow up on. And our particular questions are, how does a single point in the transporter regulate the rate of alternating access so dramatically? What defines an EMRE substrate? So the interesting thing is that our susceptibility substrates are actually mostly uncharged, whereas the resistance substrates are cationic. And then is it possible, because here we have differences in the actual small molecule that's being transported, resulting in either resistance or susceptibility. So could you design small molecules to target this transporter and for antibiotic development? And with that, I would thank the students, former and current students in BOLD who did the work that I presented today, our collaborators, and our funding sources.
Thank you, Katie. Let's start with some questions. May, do you want to do that, or should I do that?
Laurel, you do that. You're the host for Katie.
Okay. All right. Okay. So let me ask the first question, Katie. So
okay.
that correlation between the size of the substrate and the rate of interconversion was really interesting. There was this weird thing that leucine, which is hydrophobic and big, was actually very fast.
Yes. Yes, the leucine is weird, and I think it's because I don't have a figure in this talk. That's actually at a key point where the two TM3s are in close proximity to each other. And so I think there may be an issue with actual packing of the TM3 helices. If you have a leucine in that position, it may be too big to pack into that interface.
All right, next question here from Kaustup Mote. How is the dynamics in the protein and substrate taken care of while analyzing the reader data?
Oh, so we're not, we're using the point mutant that's not dynamic. So it's not interconverting. If you can, so if we run ZZ exchange experiments with S64V with this substrate bound, if we run even a 200 millisecond mixing time, we cannot detect cross peaks. So the structure experiments were done specifically under conditions to suppress alternating access, so that we did not have to worry about that. Because you're right, because we have this anti-parallel asymmetric dimer where the two halves are interconverting, it becomes impossible to really sort that out.
All right, next question from Jose Ortega. How do you think the use of mixed micelles can alter the protein dynamics?
So you have to be careful with EMRE. It's very sensitive to what detergent it's in. So dodecyl multicide is the only detergent in which it has proper substrate binding. The affinity drops off by orders of magnitude in other detergents, tenfold into DM and even more into others. We make our bicelles by reconstituting into full liposomes and then breaking them up into bicelles. And this really results in a sort of much cleaner, more stable bicell, even though these are small bicelles. So it looks like they're a sort of much more organized bicell under those conditions.
All right, Chad Rianska asked the question, how well do the amide chemical shifts agree between solution and solid preparations?
Very good. There's a little bit of offset in the amides, but the carbon chemical shifts are almost identical. And the temperature of the experiments isn't exactly identical between solution and solid. So that's probably why there's a sort of slight offset in the amides.
Marcus, how many more questions can we ask?
Keep going.
We can also go to the unofficial part, then we can ask as many as we want.
All right, let me ask.
Sounds good.
One more. For the proton F19 distance, the substrate has 4 fluorine atoms which appear identical. How do you differentiate the
so the structure calculation was done by Mei Hong's group, and what they did was to initially dock into, so there's a MD two MD simulations that have taken the crystal structure as a starting point and then done MD and explicit lipid to sort of refine that not so great structure. So the first thing they did was dock into that using pseudoatoms, and then once it was docked into that, to refine with explicit distance restraints to the fluorines that were in close proximity to the different amides.